Hello everyone, my name is William Burlew and I'm going to be presenting my project today on the testing and evaluation of pre-screening technologies for white suspicious powders. This project is being done in conjunction with the Edgewood Chemical Biological Center on APG where I work and is being funded by the Department of Homeland Security. Every year, first responders handle large numbers of cases where suspicious powders have been found in the office setting. This can be anywhere from an Office Depot to the Pentagon. Because of the large numbers of cases that are reported every year, it's necessary that there be a pre-screening technology that the first responders can use in order to determine biological activity of the powder. Currently, there are several kits that have been tested by ECBC, such as the Live Agent Test Kit, the Primer Alert, the Profile 1, and the current field-used BioCheck 2020 kit. Unfortunately, these kits are not the most effective or time-restraining or accurate kits out there because they will either take too long, or they're not as accurate, or they can be interfered with other powders or interference that can be found on the scene. So ECBC has turned its attention to the 3M Clean Trace Surface ATP reader, which has the ability to use a luminometer in order to determine the levels of ATP in a sample. Now, ATP is basically the energy inside of a cell. So if the cell is biologically active, it's going to have a large amount of ATP in the cell. And if it is devoid of ATP and there isn't any, then we know that the powder is biologically inactive. And this would be the easiest and most efficient way to test whether or not the powder is a threat to those in the office, and if any further security precautions should be taken. So there are also what we call hoax powders that can be found in these office settings, such as coffee creamer or yeast or whey powder or baby powder, depending on where the powder is found. Obviously, some powders will be more prevalent than others. However, these powders are known to have some sort of effect on tests for biological activity, so these powders must be taken into account while we are testing our new technology. This 3M Clean Trace Surface ATP reader actually currently doesn't have any forensic application. I'm not exactly sure what it's being used for currently, but I just know that it is not being used in the forensic field. But our preliminary testing has showed that this device is very accurate, is very time efficient, and we're hoping that if it proves to do better in our further testing, that we will be able to push it to the field. So in order to complete this study, we have to ask and answer some questions. So the first question we have to look into is, what is the lowest level of ATP that can be detected? Because, it's very unlikely that there's going to be like a large amount of powder just laying on this table. It's likely that it's going to be small amounts, and with the ASTM standards for collecting the powder must actually first be collected before it can be field tested. So on site, the first responders are only ever going to be testing trace material. So we're going to have to be able to determine, is there a lower limit of detection that this reader can detect at, and is it higher than what we're going to be expecting there to be on the surface still after being scraped up. The second question we're going to have to answer is how well does the device read samples that are known to have ATP in them? So basically we have to create a standard. So we should test samples that have a known amount of ATP, or if not a known amount, at least we know that there's ATP in the sample, so that we can read it and see how well the device can pick up this ATP. We can also do this by actually testing a standard addition of pure ATP, so we can see how low this limit can go before we're no longer going to see any ATP picked up by the reader. And then the third question is, how do the hoax powders interfere with the reading? So, like I said earlier, there are several different powders that can be found on the scene which can interfere with this testing. So we want to test a good amount of these powders and see how they affect different biologically active compounds and how they could mask or affect the ATP reading. It could be that, they have no effect, or they could completely mask the signal. But it's good to know because we can then, if we don't receive a signal at first, we know that we should test for hoax powders in the bulk sample later on in the lab to see if there has been any contamination of the biologically active compound with a cutting reagent. The results of this study will be analyzed to determine the ratios of false negative to positive results to aid in the evaluation of the device. So basically what this means is we're going to be testing several different samples with the device, and we're going to either know if the sample we're testing is positive or negative because we're picking out the samples. So we'll be able to create statistically significant data because we can say, okay, we've received a positive result when it should have been negative, or we've received a negative result when it should have been positive. And by determining these ratios, we can determine the accuracy of the device. So the first step in our research process is to determine the limit of detection of the device. And as I explained earlier, what we'll do is we will take a pure sample of ATP and we will dilute it down several factors and test each batch until we can no longer determine the level of ATP, and we will know that that is our limit of detection. And then we can test out different biologically active compounds to make sure that the amount of ATP in these samples is higher than that of our limit of detection, because if it's lower, then this device isn't going to be able to work at all. So then we're going to, weigh out our biologically active spores. We've decided that we're going to do 5 sets of 5 replicates each. We're going to be working with Bacillus stern, which is it's basically a cousin to anthrax, because we cannot use weaponized anthrax in our labs, but it responds equivalently to different field testings as anthrax does, so it can be used for our purposes. So basically, the stern is going to have to be processed into a usable form, and then we will be measuring out 10 milligrams of each of the stern into 25 different cuvettes, basically, that we can use to test later on. So we will take those and then we will test each individual powder by taking it out of the cuvette and dumping it onto a surface. For our purposes, we will be using office tiles that we've purchased at Home Depot. And so the powder will be dumped onto the surface. It will then be scraped away using ASTM methods in which the bulk powder is found on the tile, and then a sterile laminated card is used to scrape across the surface once and be collected into another receptacle. Then the device will be used by taking a swab that comes with the device and swabbing the area, again according to ASTM methods, where we swab from top to bottom and then left to right to make sure that we cover the entire area. And then the swab will be inserted into the device and it will be able to tell us the reading. So then after we test the biological sample of this stern, we want to see how the different hoax powders are going to affect the stern reading. So we're going to be testing whey powder, baby formula, popcorn salt, coffee creamer, all sorts of powders like that. And basically what we're going to do is we're going to mix 10 milligrams of the biologically active compound, the Bacillus stern, with 10 milligrams of the hoax powder, and it will be tested again with the device, and we can then compare the results of each mixture to the plain, just the Bacillus stern, and see which powders caused more interfering with the test. We will also be doing a toxin study. We will be using lyophilized abrin, which is basically just powdered abrin. This one's a little more complicated because of restrictions in the lab. We will be having a outside source be making the abrin, and then we will be able to take the abrin and mix it with our hoax powders and basically repeat the process that we did with the stern. A little early. So basically, after all that is done, we can test the powders by doing the false negative to positive results, because in all of our samples we want there to be ATP. So by determining how many per 25 are incorrect, we can determine the accuracy of the device. So in summary, we will be evaluating the accuracy of the 3M reader by first testing the limit of detection for ATP. We will then be doing a spore and toxin detectability study in which we will weigh out the powders and test them with the device in order to determine the effectiveness of the device. And then we will be determining the effects of interferent powders on these biologically active materials. And then we will be able to say that the data is statistically significant because of the number of replicates that we've had, and we'll be able to analyze it for the false negative to the positive ratios. And then we should be able to determine whether or not this device is suitable for field use. If you have any questions, please feel free to email me. If not, have a wonderful day. Thank you.